We started doing bioinformatics in my group just for fun, really. I never realized how useful it could be. We're interested in studying secondary structure motifs at interfaces, for instance, helices. So why do we do this? The reason is that many protein-protein interactions of biomedicinal value have recognizable secondary structures at their interfaces, like loops and helices. If you can mimic an interface helix, you may be able to displace the protein ligand and trigger an agonistic or an antagonistic response. The textbook way of making helical mimics is stapling, and that means joining two side chains together, typically by alkene metathesis. There is another way called capping. Caps are fundamentally different because they change the shape of the helix, so they terminate it and direct the chain in a different direction. We noticed that there are only good N-cap mimics, no C-cap mimics. So it was impossible to make dual-capped helical mimics. And therefore you couldn't compare them before with stapling methods. So that's where the bioinformatics came in. We knew that C-caps in helices, in proteins, are often terminated by Sherman loops. Sherman loops kink the helix out of helicity and project the side chain at the C-terminus in a different direction. There's one special residue called the C-cap, and that is the first not to have the typical phi-psi dihedral angles for an alpha helix. Besides terminating the helix, Sherman loops are thought to stabilize them as well, and that's what we wanted. Common Sherman loops are identified by a particular hydrogen bond pattern. That pattern is very similar to in a helix. It's just twisted, one residue around. We searched for that hydrogen bonding pattern in the whole PDB for every single protein-protein interaction involving unique chains. In that way, we developed a searchable database which we called Sherman Loop Finder. And suddenly, for very little effort comparatively, we can go through all this data and start to look for trends. So at one particular position, we observed, hydrophobic residues are more common than anything else. That's the C3 position. Then at the C double prime position, we observed pretty much the same thing. Hydrophobics, more common than anything else. And the same at the C cap residue. So we call these hydrophobic triangles, and they're defined by close contacts between these three residues. That led to a hypothesis. If leucines and other hydrophobics are the most prevalent of these positions, what would happen if we take them out, substitute them with cysteine residues, and then cap in a Clips reaction with this symmetrical tribenzyl bromide? In that case, the benzene rings would mimic the three leucine residues coming together to form a hydrophobic triangle. But the peptide then would be constrained in a rigid bicyclic mimic. We modeled this and it looked very good. Here's a space-filling model showing the three leucine residues and the replacement by that benzene ring. So we tried that and we called those mimics BSM, where the B stands for bicyclic, and the S stands for Sherman, and the M stands for motifs. That led us to wonder, what about the N-terminus? There are mimics for N-terminal caps, but all of them are just one ring, and they're somewhat flexible. So we began searching for nature's N caps in proteins, and they are ASX terms from aspartic acid and asparagine, and ST terms from serine and threonine. Would that bioinformatic approach lead us to similar capping at the N-terminal motif? It wasn't obvious that it would, because the N-terminus and the C-terminus of a peptide are different. And N-caps, as in ASX and ST motifs, and C-caps, as in Sherman loops, are not the same either. They have different hydrogen bonding patterns. In fact, those hydrogen bonding patterns for N caps go quite deep into the helix, and they involve two residues which we call N3 and N4. Whereas for C caps, the Sherman loop mimics, the capping motif is shallower. It protrudes more from the end of the helix. And the hydrogen bonding pattern is different. At the N-terminus, the hydrogen bonding pattern involves side chains. At the C-terminus, it doesn't. It's all main chain to main chain interactions. Nevertheless, we did find groups of 3 hydrophobic residues differently spaced at the N-terminus that are analogous to those we found at the C-terminus. Those hydrophobic triangles at the N-terminus are more squeezed, whereas at the C-terminus, they're more equilateral. So our search routine for ASX and ST motifs is on GitHub via our website. You can download dihedral angles, and you can download atom-to-atom contacts. And it turned out that at the N-terminus, the 3 prevalent residues in a hydrophobic triangle were again leucine. So would the same idea work if we made a peptide where those leucines were substituted by cysteines and then capped by the symmetrical tribenzyl bromide? Would we get an analog of the N-terminal capping motifs? And the answer is yes. So then, happily, for the first time, we were in an exciting position because we could put our bicyclic C caps at the C-terminus and the bicyclic N caps at the N-terminus in one peptide. And we could see how this would influence the conformational stability and other properties of those peptide mimics. The first thing we did was compare peptides with only an N-terminal bicyclic cap, a peptide with only a C-terminal bicyclic cap, with the dual cap mimics. And we expected the dual cap mimics would be more conformationally rigid. They were by CD. You can see this nice helical shape for the red line there. That's the dual cap compared with a linear and the C cap and the N cap controls. A really nice result came when we put these mimics into human serum and just tested the stability by HPLC. If someone injected peptide mimics as a drug, their persistence time in the serum would be critical. If they don't live for very long, they won't be very active. And look at the results. The dual cap system, that's again the red line one, is far more stable in serum than the N cap or the C cap or just a linear control. Those are degraded proteolytically much faster. And then we probed cell permeation, starting with a linear control, FITC labeled, in other words, fluorescein labeled. Not much got into the cell. It didn't light up very much. The N cap mimic and the C cap mimic permeated the cells more. And then when we examined the dual cap mimic, it was the best in the series. That conformational constraint gives us improved cell permeability. Then we made 2 more controls. We made stapled peptides with the same sequence and an I to I+4 connection via alkene metathesis. They're really what we call the gold standards for stapled peptides. How did our system compare with those? Well, you can see what's happening here. The dual cap system maintains its helical character quite well throughout the whole molecular dynamics run. However, the stapled system, which is only one ring, it's not bicyclic, remember, has C and N termini that are non-constrained. They lash around, they lose their helicity. But the really nice comparison for us came when we put the stapled mimics and the dual cap system into human serum and measured the stability. As you might expect, the stapled systems degrade from the C and the N terminus, and by HPLC mass spec, we could see the degradation products. But the dual cap system was far more stable. That's a big deal if you're using helical interface mimics as potential cellular probes and drug leads. In fact, when we looked at the degradation products from the dual cap mimic, they weren't proteolytic degradation at all. The most prevalent degradation product after a long time was oxidation of the sulfurs in the cysteine side chains. But that didn't happen very quickly at all. So in conclusion then, the prevalence of hydrophobic triangles at the C-terminus was unanticipated because Sherman loops have been known for decades. Sherman loops significantly impact the helicity, and our mimics, BSM at the C-terminus, bicyclic Sherman mimics, hold the helicity quite rigidly at that end. We needed a complementary approach at the N-terminus, and we found a set of 3 residues. We substituted them again with clips, precursors, and made BAMs, bicyclic ASX motif mimics. Again, these are quite rigid. They're not single ring systems like the previous N caps or like staple systems. Dual cap mimics have increased helicities, increased cell permeabilities, and increased serum stabilities. And that, in our opinion, is the big one, to make these mimics more bioavailable for molecular probes and drug precursors. This work was actually published in 3 papers. The last one describes the dual cap mimics that came out in JACS. Most of this work was done by an excellent graduate student, Chen-Xiong Mi, who's now a postdoctoral associate at Merck. He was helped by an undergraduate, Duyan Nguyen. She made some of the peptides, and Gavin Gao did all the cellular work. Thank you very much for listening. Bye-bye.